as a clinical physician, academic clinical physician, is there any room for interaction between us and the academic nuclear medicine community and getting involved in some of these developments or trials? So this one's working, I think. Here.
So yeah, the answer is yes. We do ISTs or investigator-sponsored trials. I have a full discovery team, at our company that is looking at other targeting motifs. And early in the development process, or even prior to getting into the clinic, it would be nice to partner with investigators that are interested, particularly in imaging, to be quite honest, because if we have target X that looks interesting from a preclinical perspective, we will have IHC and other information to help guide us on what tissues may express both cancer and normal tissues. But it's really important to get some early imaging studies. I mean, the Germans did us a great favor by running ISTs essentially. Pluvicto would never be what it is right now without them starting all of those beautiful studies. So I think the answer is totally yes. Now the question is it easier to do it in Canada, the US, or other places? Yeah, that's also something that is a bit of a hurdle sometimes, but it is totally possible.
And I would just follow up, and to get involved in the trials, so phase 2, phase 3 trials, how much of it is focused on Canada? And again, we understand it's a business situation. So is there an advantage to doing these trials in the US? Does the FDA appreciate the Canadian centers as well?
That might not be turned on there. Yeah. So yes, in fact, our trials we have open in the US as well as Canada and even in Australia. We're looking at places in Europe as well as South Africa too. So there's areas that are very productive and knowledgeable about targeted radiotherapy. And of course, we gravitate towards those sites, but those sites are increasing quite frequently. We have many of them in the States, but and that's actually where I'm located, even though I work for a Canadian company. But I think it's more. I think a lot of companies will gravitate towards doing studies in the U.S. Just because of market and everything else, and you want to establish the paradigm in the U.S. first. But that's not a given. Like I said, the Germans did fantastic. They continue to do fantastic work in pioneering new compounds. So, but for phase 1, phase 2, 3, we're going to go global.
Okay. Any questions? Well, I have one or two that have been submitted here. So Paul, what are some of the more significant challenges when it comes to translating isotope production or radiopharmaceutical development into the clinic? So from I guess, a preclinical research point of view.
Yeah, okay, well, my experience is kind of twofold. One is I participated basically in that 10-year transition of alternative technetium production. So that's a diagnostic isotope, but in that instance, I mean, between starting the effort in 2009, we received Health Canada approval for human use in 2019, effectively. So that journey was made longer by a number of hurdles, for sure. Patient recruitment domestically was one of them, hands down. But this was also an example of a problem the government was trying to solve for itself in a production method that Canada was shouldering at Chalk River, and they had mandated research groups to come up with an alternative. So it wasn't an industry pull; it was a government push, and so that was unique. And then when it comes to therapeutics, I can say with quite a bit of certainty, I mean, labs like Triumph are just not equipped to and I mean, we do have a quality program, don't get me wrong, and we certainly have enabled a decade-long imaging program with our university partners, but we are not equipped to be a production quality center. It's something we would have to hand to someone. And there's I think in Canada in particular, and I think that's the final thought I'll close my comments with, is I think every country would actually answer this question a little bit differently. And in Canada in particular, we have great government funding that goes into the fundamental stuff. We also have some great industry recipients, but there's that valley of death in between. And I don't think we as a country have really figured out how to navigate that valley yet. And that's going to be, I think, one of the challenges in the therapeutic isotope realm coming out of this effort. So that's my thought on it.
I'd like just to confirm what Paul said. I think in Canada in general, we're lacking this translational part between what is done in preclinics and then how it can be actually, pushed into clinics. And if we look at other jurisdictions, sometimes there is a tighter collaboration between academia, industry, and clinicians, and somehow it's natural. But in Canada, we need to find a way how we can make sure that this actually happens with less effort.
Mm-hmm. Yes, please.
Hi, Susie Ho, Calian Nuclear. We are an engineering firm that supports energy. I wanted to pull a thread on what Christopher was saying, and I think both Svetlana and Paul were alluding to the answer, but you'd mentioned that clinical trials using AC225 are typically performed, not in Canada, rather in the US. And can you just shed a little bit more light as to why they can't be, or they're not being performed in Canada, and what we can do to maybe bring these clinical trials home on Canadian soil?
No, great question. My understanding with working with the clinical team at Fusion is that we have several sites identified in Canada and we're working with them. It's really getting across the regulatory hurdles, and that's a slower process apparently in Canada than it is in the States. But we have every intention of opening up those sites as soon as possible. So I think Princess Margaret, other places is what I'm hearing. I'm not actively involved in those direct conversations, but it's in the workings, trust me.
Yes, sir.
Thank you very much. I have a question regarding the false positive. I mean, particularly indium labeled monoclonal antibody against insulin-like growth factor 1. Of course, before, like prostacint, etc., known to be uptaken by inflammatory lymph nodes, likely mesenteric lymph nodes, etc. Does you have this arm in not phase 2 or phase 1, or at least phase 2 arm, or not yet, or not sure?
For what?
The false positive uptake.
Positive uptake?
Yeah, false positive uptake.
Yeah. So using indium, obviously limited to spec. We do see lesions not only in lymph nodes, but also organs. I mean, it hasn't, the nice thing about PET is its higher resolution, but with indium, we still see the same lesions. So the question that we debate constantly internally is, do we need to see sub-centimeter size lesions, or do you need to just get confirmation in target lesions to enroll a patient? Because we're not going to see everything, I think, with indium. But we do visualize very nicely lymph nodes, other organs that have cancer lesions within them, even brain. So.
Hi, I'm John Porter, one of the residents from U of A. I have a question mainly for Dr. Schaffer. The actinium-225 production method you talked about the thorium spallation and the radium generator. Is this a production method that could be replicated on smaller cyclotrons elsewhere in the world, or is this something that only can be done on the 500 MeV cyclotron at Triumph?
Yep, good question. So basically, to produce radium from thorium, the spallation process requires anything above 120 MeV. And so what we're actually seeing around the world, there's probably half a dozen labs that are capable of doing something at that level, but they're not all capable of the same thing. So for the Department of Energy in the US, they have a 120, a 200 MeV machine. So their focus is to spallate thorium, and that's their primary production route. So the actinium they produce comes with that 227 component in it. Triumph is unique because it has 500, so extra kick, and it's that extra kick that gives us access to more radium. And so that's quite unique around the world. So to answer your question, The low energy cyclotron will not likely be able to use thorium-232 to produce meaningful amounts of actinium. You would have to rely on radium-226, which is I guess a cleaner transmutation, P2N, and that would be the method of choice moving forward for low energy machines. Yeah.
I just wanted to say, I think I'm going to call it to a close. I did want to have a special call out. Till now, the speakers have all been physicians, part of our broader community. I want to give an extra thanks to these three folks, two of them from the national labs, one from industry, that really took out time to come and speak to our group. So thank you very much.
Yeah, appreciate it.